Okay, so there are number of the factors we have discussed in previous lecture also, that the rate of diffusion is affected by many factors. For example, there is temperature of the media or the solution in which the molecules are moving. Similarly, we have mass of the molecule, and a larger molecule will move more slowly as compared to the smaller molecule. And we have also discussed the media in which the molecules are moving. For example, I have told you that in air the molecules, they will diffuse more rapidly as compared to the water. And similarly, the surface area. Surface area is another important factor, as we have discussed in previous lecture, that if the large surface area is available for the movement of the molecules will move more rapidly. So today we shall discuss another important factor, that is actually the distance over which the molecules are moving, the molecule will diffuse. That is another important factor, and this factor will also determine the rate of the diffusion. For example, when we talk about a cell uh, which will reach a diffusion equilibrium, and in which the molecules are moved from the blood, or moving throughout the interior of a cell, after crossing the membrane of that particular cell. So basically, the distance of the molecule from that particular cell through which the molecules are crossing through its plasma membrane, that is very much important. When we talk about the individual molecules, they move at higher speed as compared to the molecules when they are moving in a bulk, because individual molecule when individual molecule is moving in a particular solution, one important factor is that the molecule will have less number of the collisions, as there are less number of the molecules in the media, and the individual molecule will move in a straight line. So we can say that unless until this molecule collides with any other molecule in the media, this molecule will have a preferred direction of the movement, and that will be mostly in a straight line. The second important thing is diffusion times. Diffusion time increases in proportion to the square of the distance over which the molecule diffuses. Mean whenever there is increase in distance over which the molecule is moving, the diffusion time will also increase, and there is a direct correlation between the diffusion time and the distance. When the distance is increased, the diffusion time will also increase, and it will increase to the square of the distance. So let's suppose if we take the example of glucose molecule. This glucose molecule, approximately it will take 3.5 seconds to reach 90% of the diffusion equilibrium, if the point or the cell is 20 meter away from source of glucose. Mean, for example, the glucose is in a solution or in a source, and the other cell through which the glucose is actually passing, that is 10 meter away from the source of the glucose. So 90% of the glucose molecule will reach at the equilibrium stage. As I have told you in the previous lecture, what do we mean by equilibrium state? Equilibrium state actually mean when there will be no net difference on the movement of the molecule. Mean the number of the molecules when we are moving from one compartment to the other, that is equal to the number of the molecule simultaneously that are moving from compartment to compartment one. So there will be no net flux. This is what we can call it as diffusion equilibrium. So 90% of the diffusion equilibrium in case of glucose molecule will reach if the glucose is source of the glucose is 10 meter away from the cell from which the glucose is passing, and it will only take 3.5 seconds. For example, when we talk about the blood, it will uh, it will take only a very short time. But when we uh, increase the distance, it might be 10 millimeter, and it is 10 centimeters. It is not 10 meter, because this is larger distance. Meter is larger unit, and centimeter is smaller unit. So I will also reconfirm it. I think it should be 10 millimeter away, and it should be 10 centimeter away, because this is smaller unit, and when the distance is small, the molecule will move more rapidly, whereas in this case the distance is 10 centimeter. It means this is large distance as compared to this, and at this particular distance, it will take 11 years for the glucose molecule to reach 90% diffusion equilibrium. So that's how we can say that distance has a very prominent effect on the rate of the diffusion. When there is slight increase in the distance, there will be huge difference on the rate of the diffusion. So diffusion equilibrium can be reached rapidly over distance of cellular dimension. For example, nanometer or millimeter, these are the cellular dimension. But when we increase the temperature to the centimeter, the diffusion rate or the diffusion equilibrium will have a marked effect. So it takes very long time when the distance of few centimeter or more are involved. As I have told you that for the same glucose molecule to move through a cell which is which is present at a point of 10 centimeter away from the source of the glucose, it may take 11 years. And in contrast to that, when this cell is at 10 millimeter away distance, the glucose molecule will take only 3.5 seconds to pass through the membrane of that particular cell. Okay. For example, when we talk about the human being, as human is a large organism, and I have told you in previous lecture that in the body various important processes, they are actually taking place through via the diffusion process. For example, we have discussed about the movement of the oxygen, movement of the nutrients, and exchange of the gases at the blood capillaries. All these processes, they are taking place via the process of diffusion. So when we take human as an example, because human is a large organism, and diffusion of the oxygen and nutrients from the body surface to the tissue, which are actually located only few centimeters below the surface, it will be very far slow to provide the adequate nourishment. So what will actually take place in this particular case? Our circulatory system will provide the mechanism for rapidly moving material, because human is large organism. So to provide the oxygen and nutrients to all body organism, there must be a pumping system, must be a rapidly moving system that is involved in rapid movement of the nutrients and oxygen, and that rapidly pumping system is actually the circulatory system. And as all of you know that our heart actually behave as the pumping organ, and it will, once the oxygen and nutrients they are dissolved in the blood, now the oxygenated blood will be pumped to all body parts via various type of the arteries. As all of you know that the arteries, they carry the oxygenated blood from the heart to various other body parts. Can you hear me? Is there is any problem? Is there is any problem in voice?
Yes, sir.
है कोई problem?
Yes, sir. No, sir. सुन रहे हैं.
किसको problem है?
सर मुझे नाजिश को.
तो आपके नेट का इशू हो सकता है.
सर नेट का इशू नहीं, आई थिंक आपने इस वजह से.
जी.
सर नेट का इशू नहीं है, आपने रिकॉर्डिंग ऑन की हुई है तो उस वजह से प्रॉब्लम होती है, शोर आ रहा है बहुत ज्यादा.
ओके, तो रिकॉर्डिंग में ऑफ कर दूं?
नो नो सर.
Okay, the rate at which diffusion is diffusion actually move molecule within a cell, that is actually affected by the size and the distance of the cell. That's why I have told you that the size of the cell is also small. When the size of the cell is small, it will take less time for the molecule to pass through that particular cell. So that's why a cell would not have to be very large before diffusion failed to provide sufficient nutrients in its central region. That's why mostly, as all of you know, that cell they are having very small size, even in millimeter or in nanometer. So, for example, we take the center of a cell, which is I guess 20, which is having 20 millimeter diameter. So the diffusion equilibrium with the extracellular oxygen will take only 15 millisecond in this particular case, when the diameter of the cell is only 20 millimeter. But if we increase that cell, and the size of the cell is increased up to the size of the basketball, all of you know the size of the basketball. So when the size of the cell is increased to the size of the basketball, the same molecule or the same oxygen molecule will take 265 days to reach the equilibrium. So it means for the rapid diffusion of the molecules, whether they are those molecules are oxygen or the nutrients, the size of the cell should also be small. So it will take small time for the molecule to diffuse to that small size cell. Okay. Similarly, how we can actually measure the rate? We can measure the rate of the diffusion by measuring the rate at which the intracellular concentration approaches the diffusion equilibrium. As I have discussed in previous lecture, that for example, when we take the example of the glucose molecule in the example we have studied in the first lecture, all of you know that both the compartments, they will reach the diffusion equilibrium when there is equal concentration of the glucose in both compartments. So in the similar way, we can, so it means when there is equal concentration of the glucose in both compartments, it means the rate of the diffusion is actually 10 millimole per liter, and at particular time. For example, we have said that time C or time A or time B. So by monitoring the intracellular concentration of the molecule, of the diffusing molecule, we can also measure the rate of the diffusion. Let's suppose initially the concentration is 20 millimole per liter, and after certain period of the time, the concentration in the other compartment is 5 millimole per liter. So automatically we can measure that 5 millimole per liter glucose molecule, they have diffused from the compartment 1 to compartment 2. That's how we can measure the diffusion rate by monitoring the intracellular concentration of the molecule in the other cell, in which through which the molecule is actually diffusing. So when we talk about the extracellular fluid, extracellular concentration of the molecule is always large. Let me show you the previous slides which we have discussed in first lecture. Okay, can you see the slides? Can you see the slides from the previous lecture?
Yes, sir.
Yes, sir.
I want to just share a point with you. Okay. If you look at this particular example, here you can see that extracellular concentration of the glucose molecule is very high as compared to the intracellular. Or for example, this is concentration of the glucose molecule in C1, and this is concentration of the glucose molecule in the C2. So what I have said you that how we can monitor the rate of the diffusion. We can simply monitor the rate of the diffusion by monitoring the number of the molecules uh, uh, that has actually traveled from the compartment 1 to compartment 2, because if we start to count the number of the molecules which have actually which have been lost from this compartment, it is very much difficult because here the concentration of the solute is very high. So we can simply measure the rate of the diffusion by counting the number of the molecules that have been added to this compartment. So that was the point which I want to make it clear for you, that because the extracellular concentration of the solute or any other molecule, for example, when we talk about the oxygen or any other molecule which is present in the extracellular concentration, there will always be very high concentration of that molecule in the extracellular side. So we can't measure the rate or the number of the molecule that has actually that have passed through the membrane. We can measure the rate by counting the number of the molecules that have moved from extracellular side to intracellular side. So that's how we can calculate the rate of the diffusion. Another thing which I want to correct here, in previous lecture.
सर आपकी आवाज नहीं आ रही।
मुझे भी नहीं आ रही है सर।
Can you hear me?
यस
Can
सर
you
आप
hear
ठीक
me
है।
now? Okay, sorry.
यस सर।
Okay. I want to clear this point, as I have said you, as I have said that we can calculate or we can measure the rate of the diffusion by measuring the intracellular concentration of the molecule. Why? As in this particular figure, you can observe that in the extracellular side there is a very huge concentration of the solute particle. So we can't simply measure the number of the molecules that are lost from this area. But on the other hand, we in this area we can measure the number of the molecules that are added to this particular region. So that's why we can say that we can measure the rate of the diffusion by measuring the intracellular concentration of the solute, because normally the solute are present at very high concentration on the extracellular side. So it will have no obvious effect when some molecules they have moved from extracellular side to the intracellular side, but it will have definite effect on the intracellular side because the concentration of the solute will increase to the intracellular side. So that's why a rate of the diffusion can also be measured by monitoring the intracellular concentration of the solute, that how many solute particles they have actually moved from the extracellular side to the intracellular side. And another point which I want to correct here, I have said in previous lecture that in this graph, for example, I have said that time is along the y-axis. It is not along the y-axis. This is actually the x-axis and this is y-axis. So time is along the x-axis and glucose concentration is along the y-axis. So you may correct this as well. Okay. Now going to today's lecture. Okay. So here the rate that can simply be measured by monitoring the intracellular concentration of the solute, and why this rate can be measured easily, because on the extracellular side the volume or the concentration of the solute is very large, and even after losing some solute particle from the extracellular side, still there will have no obvious effect on the solute concentration on the extracellular side. It will remain more or less, it will remain constant. So that's why we can measure the diffusion rate by monitoring the intracellular concentration of the solute. So net flux of the material across the membrane, that will always be from region of higher concentration, which is actually the extracellular site in extracellular solution, to the region of lower concentration, which is the intracellular fluid. And I have told you in previous lecture that the net flux is actually indicated, or it is actually determined by the arrowhead. The arrowhead will determine the magnitude of the net flux, as well as it will also determine the direction of the net flux, whether the molecules they have moved from 1 to 2 or they have moved from 2 to 1. So the length of the arrowhead will determine the magnitude, and the arrowhead similarly at the same time this will also determine the direction of the flux. So magnitude of the flux is directly proportional to the difference of the concentration. As we have already discussed that we can calculate the net flux by having the difference of the two individual fluxes. So this is actually the difference of the concentration across the membrane. For example, C naught is the extracellular concentration, and it will remain same. Here you can also observe in this graph that the extracellular concentration of the solute will remain same, because there is very huge concentration of the solute, large number of the solute particles they are present on the extracellular side. So moving a few solute particle will have no obvious effect on the solute concentration on the extracellular side, but it will have definite effect on the solute concentration on the intracellular side. Mean at this particular point there is same concentration of the solute, whereas similarly at this point there is same concentration of the solute on the extracellular side, but at this point there is zero concentration of the solute, but at this point there is definite concentration of the solute. There are definite number of the solute particles that are present on the intracellular side. So that's how the extracellular concentration will remain same, whereas the intracellular concentration will increase, and a point will reach when there will be equal concentration of the solute on the both sides. That is Ci, which Ci is actually the intracellular concentration of the solute, and C naught is the extracellular concentration of the solute. So at equilibrium stage, Ci will be equal to C naught, and we can also calculate this with the help of a mathematical equation. So the magnitude of the net flux is directly proportional to the difference in the concentration across the membrane, C naught minus Ci, and the surface area of the membrane, which is actually determined by the letter A, and the membrane permeability constant, which is Kp. Kp is the membrane permeability constant. So F is the magnitude of the net flux. So we can simply calculate the magnitude of the net flux as F is equal to Kp A into C naught minus Ci. You may write this equation on your notebook, that this F is actually the magnitude of the net flux that can be calculated as F is equal to Kp, where Kp is the permeability constant of the membrane. Whether the membrane is selectively permeable, it is impermeable, or it is totally permeable. So Kp is the permeability constant, where A is the area, and C naught is the extracellular concentration minus Ci, whereas Ci is the intracellular concentration. So this is actually the numerical value, the mathematical expression of the magnitude of the net flux. So when we talk about this permeability constant, which is Kp, this is experimentally determined number for a particular type of the molecule, at a given temperature. At a given temperature, every molecule can have a specific permeability constant, and it actually reflects the ease with which the molecule is able to move through a given membrane. For example, when this Kp permeability constant is high, it means the membrane is almost permeable to the molecule through which the molecules are passing. And when this Kp is having value which is less, it means the membrane is less permeable, or partially it is permeable to the molecule. And when this Kp value, we have Kp value is 0, it means the membrane is totally impermeable to molecules which are actually trying to move across that membrane. So this permeability constant, it is a numerical value, experimentally determined value, and it actually indicates, it actually represents the feasibility for the movement of the molecule across the membrane, and it is determined by a number. It will have a value, whether it is 1, less than 1, 0.5, or any other number. So here we can also see in this figure that when there is increase in the intracellular concentration, as the substances, they will diffuse across a membrane, the intracellular concentration will increase, and the extracellular concentration will more or less remain constant, and a point will reach when these two lines, they will merge with each other. At this particular point, the intracellular concentration will be equal to the extracellular concentration. This is what we call this as diffusion equilibrium. In other words, if we talk about the permeability constant, as I have told you that if the permeability constant is having a greater value, the flux will automatically be large, and it will allow the large number of the molecules to cross across that membrane, and definitely the net flux will also be greater in this particular case. And the rate at which the molecule diffuses, they diffuse across a membrane, that is measured by their permeability constant. And when we compare the rate of the molecules which are moving across a membrane, they are 1,000 times smaller than the rate, diffusion rate, when the molecules, the same molecule, when that is moving through a water layer of the equal thickness. So it means the composition of the membrane matters. It means if the membrane is having the water layer and the thickness of the two membrane that is same, then the rate of the diffusion, there will be huge difference in the rate of the diffusion. So membrane actually act as a barrier. It actually slows the diffusion of the molecule across its surface, or sometime the major factor which actually limits the movement of the molecule, which actually affects the diffusion rate across the membrane, that is lipid bilayer. As all of you know that membrane is made up of the lipid bilayer. That's why we can say that the plasma membrane is actually selectively permeable membrane. It will allow the lipid soluble particle to move through easily, whereas it will reject the movement of the water soluble molecule. So when the permeability constant, when we take the permeability constant of different organic molecules, and we examine, we check their permeability constant in relation to their structure, there will be a correlation. Mean the permeability constant value will have the definite effect on the diffusion of the molecule across a membrane, and similarly the structure of the molecule will also matter. When we talk about the structure with respect to their composition, with respect to their size as well, but majorly the structure of the molecule, when we take the structure of the molecule, we will normally, or we will most of the time, we will consider the composition of that molecule, whether that is polar molecule or that is a non-polar molecule. So as you all of you know that polar molecules are the water soluble molecule when they are moving across the membrane, because the major portion of the membrane is lipid bilayer. So that's why the polar molecule, they will have difficulty while diffusing across the membrane. Whereas when we take the non-polar molecule, for example, when we take the lipid soluble molecule, they will move more rapidly across the membrane because of the composition of the membrane. So two factors they are actually important in this case. One is the composition of the membrane through which the molecules are moving, and the other is composition of the molecule itself, whether they are lipid soluble, whether they are water soluble. So if the molecules are water soluble, they will have difficulty while crossing through the membrane, whereas when the molecules are lipid soluble, they can easily cross across through the membrane because of the composition of the plasma membrane. So lipid soluble, for the lipid soluble molecule, the plasma membrane will have the large Kp value. It means it is highly permeable to the lipid soluble molecule, and on the other hand, for the water soluble molecule, the plasma membrane will have the small Kp values. So that's why non-polar molecule, they will only dissolve in the non-polar region of the membrane, and those non-polar regions are occupied by the fatty acid chains of the membrane phospholipids. And all of you know that membrane is actually composed of the it is lipoproteinaceous layer in which their lipid is present in the form of the bilayer. When we talk about the polar molecule, because of their composition, the polar molecule will have lower solubility in the membrane lipid, and it will affect their diffusion rate as well. So when we increase the lipid solubility of the substance by decreasing the number of the polar or the ionized group it contains, you must know the polar molecule. Polar molecules are those molecules that can be dissociated in the ions. For example, when we call the water as polar molecule, it means water can be dissociated into hydrogen ion plus hydroxyl ion. So all those molecules which can be dissociated into the ions, they will be called as polar molecule, and they will have less permeability constant across a plasma membrane. So when we decrease the number of the ions, their solubility across the membrane, that can be increased. So that's how the polar molecule, their solubility can be increased by decreasing the number of polar or ionized group it actually contains. So oxygen, carbon dioxide, fatty acid, and steroid hormones, they are example of the non-polar molecule, and they will diffuse more rapidly through the lipid portion of the membrane. And when we talk about the organic molecules, for example, they are basically ionized at the polar molecule. So these polar molecule, they contain the ionized phosphate group. So when they have any ionic composition in their structure, they will have difficulty while crossing the lipid bilayer. So most of those substances which are polar, which are having any ionic composition, they will be retained by the cell, and they will not be able to move through the lipid bilayer, because the lipid bilayer will actually act as barrier for those polar molecule. So when we look at the ions, for example, so now the question arises now that how the polar molecule or the ions, how they will move. As we have said that non-polar molecule, because of their composition, they will have large permeability to cross the plasma membrane, so they will move more rapidly as compared to the non-polar molecule, and non-polar as compared to the polar molecule. And polar molecule are actually they are having the ions. So how these ions will cross the membrane? So when we take certain ions, for example, we have sodium ion, potassium ion, chloride ion, and calcium ion. They diffuse across the membrane at rates that are much faster than that would be predicted from their very slow solubility in the membrane liquid. So although we have already said that the molecules or the compound having ionic composition, they will have very slow solubility, but when we take the sodium, potassium, and chloride and calcium ion, their diffusion rate is very high across the membrane. So how that is possible? There are, in addition to the lipoproteinaceous layer, the plasma membrane that also carries specific type of the channels, and through these channels the ions, they may move more easily at a very higher speed, at very higher rate. That's why these molecules, they are having higher diffusion rate, because of the presence of the certain ion channels which are present on the surface of the plasma membrane. So different cells have quite different permeability to these ions, and non-polar substances, when we talk about the non-polar substances, they have similar permeabilities when different cells are compared. When we compare different cells, because all of those cells, they will have more or less similar composition of their membrane. So all those cells, they will have similar permeabilities to the non-polar substances, because non-polar substances will easily cross through those cells. But when we talk about the polar substances, and when we specifically, when we take these ions as an example, the different types of the cells, they will have different permeability to these ions. And how this is actually possible? This is possible because of the presence of the number of the ion channels on the surface of the plasma membrane. And these ion channels, they are variable in different cells. For example, if we take two cells, one cell may have large number of the ion channel for the sodium ion, whereas the other cell may have large number of the ion channel for the potassium cell. So these two cells have the different permeability for these two different ions. But when we take the non-polar molecule, and when we calculate their permeability, in case of these two cells, both these cells, they will show similar permeability to the non-polar molecule. So actually the number of the ion channels which are present on the surface of the different cells, on the membrane of the different cells, that is variable, and that's how the permeability to these ions, that is also variable. So when we take artificial lipid bilayer, and that is containing no protein, they will be impermeable to the ions. It means that movement of the ions are actually accomplished, are actually facilitated by the protein component of the membranes. For example, if we take an artificial membrane and we remove all the protein component from the surface of that particular membrane, and we carry out the diffusion of the ions, for example, sodium ion, potassium or chloride ion. So in real sense, these ions, they will not be able to move through the membrane because of the absence of the protein component. So it is actually the protein component of the plasma membrane or of the cell membrane that facilitate the movement of the ion across the membrane. So as all of you know, that there are certain types of the proteins which are present on the surface of the plasma membrane, and these proteins can be one of those protein can be called as integral membrane protein. Integral membrane proteins are those proteins that can span through the lipid bilayer. This is lipid layer. So these are lipid bilayers. Similarly, these are lipid bilayer, whereas these are the proteinaceous structure, and these are called integral proteins. These are called integral proteins. They can span across the surface of the membrane, and they can span through these lipid bilayer, and they form the various helical structure, and those sometimes those are called channels. These proteins are actually forming the channels, and those channels are ion channels, and through these ion channel, the sodium ion or the potassium ion or the calcium ion, they will move. So the integral proteins which are present on the surface of the membrane, and they can span through the lipid bilayer of plasma membrane, they will form the channels, and through those channels the movement of the ions that can be accomplished, and ion can diffuse across those channels. Single protein, it can have a conformation similar to that of the donut. Donut mean any structure having a lid that is capable of opening and closing, that is called donut. With the hole in the middle, as all of you know, the structure of the donut. So these channel protein, they are actually behaving as the donut, having a central hole in their center, and that hole can be open or and it can be closed at certain period of the time, and it will facilitate the movement of the ion across a membrane. So this is the structure. This is model of an ion channel composed of 5 polypeptides. These are 5 polypeptide subunit, and channel subunit, it is having an integral membrane protein which is carrying 4 transmembrane segments. These are transmembrane segments, 1, 2, 3, and 4. These are this part is integral part, and this part is transmembrane segment. So this integral protein is having 4 transmembrane segments, each of which is having alpha helical configuration. As all of you know about the structure of the protein, that protein can be arranged in the form of alpha helix, or it can be arranged in the form of the beta sheets, or in the form of the beta turns. So in this particular example, these integral proteins, they are forming the alpha helical structure. They are having 4 subunits, and those subunits are actually transmembrane protein. They are traversing across the surface of the lipid bilayer. So this model has only 4 transmembrane segment. Some can have as many as 12 or many, but in this particular case, this is having only this protein is having 4 transmembrane segments. Okay. Now moving on to the next slide. This is another subunit of the protein, the same subunit. This is the 3-dimensional structure of the membrane protein that is present within the membrane, and it is again having 4 transmembrane helix which are aggregated together. In this case, these 4 transmembrane 1, 2, 3, and 4, they are aggregated, and this is the integral part of the protein, and this is the transmembrane part of the protein. So diameter of the protein channel are very small, as it will allow the movement of the small ion through it. So small size of the channel, it will prevent the larger organic molecule from entering the channel. So ion channel is having the selectivity for the type of the ion. For example, we have sodium ion channel. It will only allow the movement of the sodium ion through it. Or we have potassium ion channel. So potassium ion channel will only be permeable to potassium ion, and it will be impermeable to the sodium ion. Similarly, if we have chloride ion channel, it will be only permeable to the chloride ion, whereas it will be impermeable to any other ion, for example, sodium, potassium, or calcium. So this selectivity is actually determined by the 2 factors. One is diameter. Diameter means size of the ion channel or size of the channel protein, and similarly the surface of the protein subunit that form the channel walls. And this protein will electrically, it will either attract or repel the ion. So two factors, they determine this selectivity, this permeability. One is the size of the ion channel. The size of the ion channel should match the size of the ion. Mean the ion channel should have the size that should facilitate the movement of the ion, and similarly the electric composition of the channel protein, it will attract certain ions and it will repel certain other ions. So for example, when we look at the potassium channels will only allow the movement of the potassium ion, and they are specific for the movement of the potassium ion. Similarly, sodium channels, they are specific for the movement of the sodium, and still there are certain other channels which are also present. They are called sodium potassium channels. So when they are present in combination, so it means these sodium potassium channel, they will allow the movement of both sodium and potassium through themselves. So that's how we can say that these ion channels, they are actually showing the selectivity. Here is another example of these ion channels. Again, there are 4 subunits. So the ion channel consists of 5 of the subunits. In this case, here are 5 subunits in this case. So helical transmembrane segment, it is having transmembrane segment, which is basically this light purple in structure. Of each subunit, it forms the sides of the channel opening. This transmembrane segment is forming the sides of the channel opening. And similarly, presence of the ionized amino acid side chain along this region, it actually determines the selectivity of the channel to the ion. Mean it is having the amino acids, and those amino acids they are having positive charge and negative charge, so they will determine the selectivity of the ion channel to the ion. So this model show the 5 subunit, although they are identical. Here in this particular figure, many ion channels, they can be formed by the aggregation of the different subunits, different polypeptide subunits. In this, all these ion channels, the channel protein, they are formed of the same polypeptide subunits, but the possibility is there that in certain other cases, the ion channel can be formed by the aggregation of different polypeptide subunits. So diameter of the protein channel, as I have told you, that the selectivity is determined by the size of the protein channel, and the diameter is also very much important. So diameter of the protein channel is very small, and it will be slightly larger than the size of the ion. Ion have the smaller size than the ion channel protein. So the ion channel protein will have slightly larger diameter, a slightly larger size as compared to the ion which is actually passing through them. Small size of the channel, it will provide the selectivity to those channel protein. It will prevent the larger polar organic molecule to enter through those channels. So the selectivity is actually determined by the size and composition of the channel protein. Ion channels show selectivity of the type of the ion that can pass through them, and selectivity is based on 2 factors. One is the channel diameter, and the other is composition of the channel protein, whether the channel protein is carrying the charge or the polar surface of the protein subunit that form the channel walls, and these charge or the polar surfaces, they will attract or repel the ion. For example, if on the surface of this channel protein the negative amino acid, negatively charged amino acid are present there, so this channel protein will attract the positively charged ion. For example, it will attract the sodium ion, or it will attract the potassium ion, and it will repel the chloride ion. Similarly, if the surface of this membrane channel protein is carrying the negative charge, positive charge, sorry, it will repel the positively charged ion. For example, sodium ion and potassium ion or the calcium ion will be repelled by this channel protein, whereas it will attract the negatively charged chloride ion. So channel diameter and composition of the channel protein, that is also very much important in carrying the ions across the membrane. So that is all about for today's lecture. You may ask the question now. सर खरीद ठीक है, मैं क्लास में।